Good morning, and thank you, Bob, for organizing this. It's a very worthy cause. Heart attack is a condition that afflicts 1.1 million Americans every year. And what happens is that one of the arteries that feeds the heart, a coronary artery, becomes occluded with a thrombus, with a blood clot that forms usually on top of a cholesterol-laden plaque. And when that happens, basically part of the heart muscle is starved and turns into scar. Now, most of what we do these days as cardiologists to treat myocardial infarction or heart attack has to do with promptly reopening the artery with a balloon catheter. And that actually works very well, but about a quarter of the patients, so about 300,000 patients a year, are left with pretty large scars that don't go away on their own and that involve more than 20% of the heart muscle. So overnight, 20% of living heart muscle, about 1 billion heart cells, turn into scar and die, and what's left is a heart that can't adequately pump to meet the body's needs, and often these patients go on to develop heart failure. And heart failure involves an inability to exercise and shortness of breath. And these patients have a higher mortality in general than many with malignant tumors. So what we'd like to do is actually to challenge dogma, which teaches us that once formed, scar is there forever, and that healthy heart muscle, once lost, doesn't regrow on its own. So we'd like to use cell therapy to achieve genuine regeneration in the injured heart. Regeneration, if you actually think of it from first principles, and here's a definition from the American Heritage Dictionary, is regrowth of lost or destroyed parts or organs. And it turns out that in the heart there is a transient imaging method, magnetic resonance imaging, with a contrast agent that can tell you quantitatively the difference between scarred muscle and living heart muscle. You can actually put a number of grams on how much of your heart is turned to stone and how much of your heart is still living. And so if you use that as a method of trying to benchmark, how would you assay how and whether you have succeeded in achieving true regeneration? The criteria would be as follows. The first one would be growth of new, healthy cardiac tissue. If you go from, let's say, having 100 grams of healthy heart tissue and 40 grams of scar, and after the end of a clinical trial, you have 120 grams of heart tissue and 10 grams of scar, that's a huge success. You've taken that patient from a high-risk group that's likely to go into heart failure and die prematurely to a low-risk group that has every chance of living a normal life. That tissue should have improved function because if it's healthy, in fact, it should contract more vigorously. The reduction of scar per se is a bonus. It's not necessarily part and parcel of regeneration, but if regeneration happens by converting scar to living heart muscle, that would be a huge bonus because less scar is good, more living heart muscle is even better. So we developed a technology when I was at Johns Hopkins and then brought it here to Los Angeles in 2007 when we came. And all the clinical trials have been done in Los Angeles. And it's called the CDC technology, or Cardiosphere-Developed Cell Technology. And the idea is to try to harness, in a very simple way, the heart's endogenous healing power by taking the cells out of the heart that normally mediate the wear and tear of the heart. And in all of the normal hearts, and even in disease hearts, there's some effort to try to replace wear and tear, but it amounts only to about 1% per year. So when you lose 40% of your heart cells overnight in a catastrophic heart attack, you can see how that endogenous capacity might easily be overwhelmed. But our approach was to try to take the cells out of the heart that mediate this normal wear and tear repair and put them back into the zone of injury and see if we could reverse the equation. And so we came up with this mechanism to take individual small samples from patients and grow them up in the laboratory and put them back in, starting with a very small sample of heart muscle taken with a biopsy. That was back in the days when we thought that perfect matches were absolutely essential, so we didn't want any immunological complications. We now know that these cells are very interestingly immunomodulatory and sort of protected from rejection, and we can actually use allogeneic cells, off-the-shelf cells derived from donor hearts, and that's in fact a discovery that we've made using CIRM funding. But the first clinical trial involved perfect match autologous cells, And we ascertained patients who had a heart attack, harvested their cells, and we could get about 30 million of these CDCs in about 3 to 5 weeks. And our 3 to 5 weeks. And our target dose for therapeutics was 25 million. So we did the Caduceus trial. The Caduceus is the symbol of medicine, which is shown on the upper right corner on the slide. And the results were published in The Lancet last year, and the complete results after 1 year of follow-up were published just recently in the Journal of the American College of Cardiology. So what in order to get into Caduceus, you had to have had a heart attack within 30 days of screening, and we didn't rush to treat patients right when the heart attack happened because we knew we would take 3 to 5 weeks to grow the cells anyway. So there was no reason to rush to therapy. We allowed them to quiet down, and then we could very reliably ascertain who was at high risk and who was at low risk for progression or heart failure. And the patients had to have some degree of left ventricular dysfunction. Their hearts had to be pumping somewhat weakly with an ejection fraction of less than 45%, the normal being probably 60% or more. And we then qualified the patients and fully randomized them either to receive standard of care, those are control patients, or in a 2-to-1 manner that had twice as likely a chance to get into the treatment arm. And the patients who were treated ended up being 17 patients, and they underwent a biopsy and reinfusion of their cells down the infarct-related coronary artery using a standard minimally invasive percutaneous technique. And all patients, whether they were controls or cell-treated, underwent the same phenotyping. They underwent the same testing to see if they were getting better or not, and the primary modality to assay efficacy and safety to rule out tumors, for example, was by magnetic resonance imaging. I'll show you some of those results. And so what we were very excited to find is that this was the first therapeutic modality ever reported in a controlled clinical trial that decreased scar in a setting where scar was thought to be irreversible, but more importantly, increased viable tissue, thereby fulfilling the criteria for therapeutic regeneration. These are magnetic resonance images in a patient in the top row at baseline, 88 days after he had had a pretty massive heart attack, and 6 months later. And you can see what looks like a concentric circle there should be all black. A healthy heart would be a black donut in this image, and in the upper part of the image where the arrows are, much of the black is replaced by white, and that's scar within the heart. And you can see in the front of the heart, the top part, the thickness of the scar actually goes all the way through the heart muscle and extends the circumference about 40% of the heart muscle. This person had lost about 30% of their functional heart muscle. And 6 months later, there's less white, less scar, and more black, which is living heart muscle tissue. This is just one slice. With MRI, we can volumetrically, systematically reconstruct and actually get a number for the amount of mass that's turned into scar or that's living heart muscle. So you can see the patient who got treated with cells looks like they got better by this criterion, whereas the patient on the bottom, who was imaged 89 days after a heart attack and then 6 months later, but received standard care, did not have any shrinkage of his scar, nor was there any apparent increase in the amount of living heart muscle. One extremely important feature of this study is that the control patients, the ones that were randomized to standard therapy, fulfilled all the pessimistic expectations that we were taught in medical school. On these bars on the left, you see the change in scar mass at 6 months and at 12 months from baseline, and the values are clustered around 0. So that means that they didn't get better spontaneously. Scar, once formed, did not go away on its own. On the right-hand side, the bars show the changes in viable mass, the change from baseline to 6 months or 12 months in how much of the heart was occupied by healthy heart muscle. Again, clustered around 0, showing that heart muscle, once lost, doesn't regrow on its own. And then if we look at the data from the patients who had received their own cardiosphere-derived cells, at 12 months, there's a shrinkage of about 12 grams in the amount of scar mass. That's a huge effect, because these patients started off on average with about 25 grams of scar. So in and of itself, that's a big effect, but what really got us excited was the fact that these patients regrew on average at 12 months 22 grams of myocardial mass, of healthy, functioning muscle mass that had been thought to have been lost forever. And that is an enormous effect. It's like It's the mass of 500 million new adult cardiac myocytes, and these are patients who have lost about 1 billion cardiac myocytes. This is important for CERM. It's important conceptually for medicine because it's the first evidence in any controlled clinical trial of therapeutic regeneration. A person can come in and lose half their liver due to a motorcycle accident. It'll regrow on its own. A salamander you can cut off his tail or his foot and it'll regrow the tail or the foot. But there is nothing that has ever been done to a human being to cause something to regenerate that has been thought to be lost forever before this clinical trial. And it shows that cell therapy can work, and it can work incredibly well. And the tissue is more contractile. It's beating more strongly in the region of the injury. It's thickening more, and the thickness of the wall is higher, which is actually a very valid therapeutic principle. Now, I told you that we subjected each one of these patients to a biopsy and then gave them their own cells. And that's beautiful from the immunological point of view, but that becomes kind of a cottage industry in that every single patient has to be treated individually. There are timing constraints, 3 to 5 weeks. If we wanted to treat a patient acutely right at the moment of injury, that wouldn't be possible. It's expensive. It's risky because the patients have to undergo an invasive harvesting procedure, and there's not a 100% success rate with growing the cells from an individual biopsy. We had a success rate of 91%. With practice, we could probably get it up to 95%, but still, who wants to undergo a biopsy with a 5% risk of manufacturing failure? There's suboptimal quality assurance and quality control because every run needs to be done basically as an individual run instead of doing a master cell bank and creating, let's say, 6,000 doses at one time. There's interpatient variability and cell potency because no matter how hard we try, we can never separate the patient from his disease. So if it was a diabetic who's 72 years old, and had a history of bone marrow dysplasia, those cells are going to bear an imprint of that. They're not going to be like cells would be from a healthy donor that maybe died as a result of a motor vehicle accident and whose heart was donated for making cells because it couldn't technically be used for transplantation for some reason. So we wondered whether allogeneic off-the-shelf therapy was safe and effective, and we did studies in rats and in pigs funded by CERM to validate the principle that indeed completely mismatched cells work just as well as perfectly matched cells, even though they're immunologically cleared. But within 4 weeks, the effects last upwards of 1 year. So these are data from a pig that was done as part of the CERM disease team 1, the first CERM disease team to actually lead to a validated therapeutic candidate in a clinical trial. And they basically show exactly what I'd shown you in the humans, less scar and more working heart muscle. And you can see on the bottom, on average, that the scar size gets smaller in the treated animals. It doesn't change in the controls. The viable mass increases in the treated animals, does not in the controls, and the ejection fraction, which is the global function of the heart, is much better preserved in the treated pigs than in the controls. Using these kinds of data, we were able to go to FDA and IRB and get clinical trials started for testing these cells. But before I describe those clinical trials, is the last thing I'm going to do. I just want to summarize and tell you that perfectly matched autologous cells produce therapeutic regeneration in humans post-MI. That's the Caduceus work that was published in Lancet. That allogeneic CDC, off-the-shelf CDCs, without immunosuppression, are safe, and they promote cardiac regeneration and improve function via stimulation of endogenous repair. And so we now are motivated to test off-the-shelf products clinically to see if we can effect cardiac regeneration without the need for biopsy using banked products. And these are 2 clinical trials that I'm going to describe. And they take advantage of a manufacturing process that's been reduced to practice by Capricor, which is a biotech company based in Los Angeles, of which I'm a founder, and which now benefits from CERM funding. A donor heart that is technically unsuitable for use for transplantation, for example, because it has a local contusion, so a surgeon doesn't want to put it into a patient, but we can use the 90% of the heart that's uninjured to grow stem cells, and it's transplant quality, free of viruses, et cetera. We dissect that, create a master cell bank, and we can generate up to 6,500 doses per heart, even on a relatively non-scaled up model, which is what we're working on now. We can do QA and QC until the cows come home because we've got all day to prepare the doses, but then the doses can be frozen and thawed and delivered to patients for immediate use. And the first clinical trial using this allogeneic cell technology is ongoing. It's called All-Star, Allogeneic Heart Stem Cells to Achieve Myocardial Regeneration. It's a CIRM-funded trial. The principal investigators are both at Cedars-Sinai, Tim Henry, who's the chief of cardiology, and Raj Makkar. And in order to get into All-Star, you have to have similar entry criteria to Caduceus. You had to have had a heart attack with a fairly large scar and some dysfunction of the heart. And now we're actually treating patients up to 1 year after the heart attack to see if it makes a difference, whether we capture them in the first 3 months or whether we can let them go more chronically. It's a double-blind and placebo-controlled trial. We as cardiologists are extremely persnickety about making sure that the quality of the evidence is outstanding, and so we tend to favor trials in which neither the patients nor the doctors know which treatment group they've been assigned to until the code is broken at the end of the study. There's an exception in the sense that in phase 1, when we're dosing the patients to see if the product is safe and whether the dosage is adequate, both the patients and the doctors know what they're getting. And we have here today actually Dr. Subiya Subramanian, who's a Stanford professor who is in the Internet Hall of Fame and a serial entrepreneur who actually was one of the phase 1 patients in All-Star. And he'll tell you about his experience. The larger trial, the phase 2 trial, which is just beginning, will involve 20 sites in the US and will recruit a total of 274 patients. And so we'll be able to get answers from this with allogeneic CDCs. And we actually think that there's a fairly good chance that this will be the first CIRM-funded cell product that will go to market if everything goes well. At least it has a good shot of that. And then we're very excited, too, about another trial that we've gotten FDA approval for and IRB approval for and are poised to start. It's called the DYNAMIC trial, Dilated Cardiomyopathy Intervention with Allogeneic Myocardial Regenerative Cells. And the idea there is to target patients with really, really severe heart failure. These are patients that have a 40% chance every year of either being rehospitalized or dying. So if we impact on that even a little bit, we can save lives measurably with a relatively small trial. And this trial, which I think will be exceedingly important for public health, is now under consideration at CIRM for funding as a disease team 3 trial. And I'm hopeful that that'll happen because without this funding, the trial will not happen. The NIH maybe will give you $250,000 a year to test something in vitro or in mice. It's exceedingly hard to even get like a 10-patient trial, but you would never, ever, ever be able to get a 48-patient study out of NIH. It's just not going to happen. So with this as a backdrop, I'd like to introduce Professor Subramanian.